Welcome to The Explainer. I am absolutely thrilled to have you here today because we are embarking on a massive microscopic journey. We're zooming all the way in to look at the very foundation of our existence, the fundamental, structural, and functional unit of life. Yep, we're talking about the cell. Get ready because we are literally about to decode the incredible machinery that is keeping you breathing, thinking, and just living at this very second.
But to start us off, I want you to consider a real macro-philosophical question. What exactly is the boundary between living and non-living? You look at a piece of plastic or a rock and, well, clearly non-living. Look at a bird, a plant, or yourself, clearly living. But what fundamentally separates the two? The answer, absolutely unequivocally, is cellular organization. If you want to be considered alive in the biological world, you've got to have at least one complete cell. It's the definitive proof of life.
Okay, let's dive into this. Here is our roadmap for today's explainer. The definition of life, formulating the cell theory, the universal cell components, plant versus animal cells, prokaryote versus eukaryote, and finally, a quick tour inside the bacterial cell. So, Section 1, the definition of life. We just established that having a cell is the defining feature of life. But why is a complete cell absolutely required to survive independently? Like, why can't we just have pieces of a cell floating around doing their own thing? Well, it all comes down to cellular dependency. Think about the mighty mitochondria, the classic powerhouse of the cell. It can make all the ATP energy you could ever need, but it can't just do it out of thin air. It needs fuel. It needs food. And where does that food come from? The chloroplast. But the chloroplast only knows how to make the food, not the energy. And honestly, neither of them knows when to do their jobs without instructions from the nucleus, which acts as the brain. So you see the issue here? These components are completely codependent. Anything less than a full, complete cell simply cannot survive on its own. It absolutely requires the whole synchronized team. Moving right along to Section 2, formulating the cell theory. You know, how did humans actually discover these invisible little units of life? It obviously didn't happen overnight. It took some brilliant minds, and crucially, the invention of the microscope. Look at this rapid historical progression. It all kicked off in 1665 when Robert Hooke first observed dead cork cells. Then, fast forward to 1831, and another Robert Brown, peered even deeper and discovered the dense central nucleus. And things really heated up in the late 1830s. A German botanist named Matthias Schleiden realized that all plants were made of cells. Just a year later, a zoologist named Theodor Schwann looked at animal cells, noticed they had a thin outer plasma membrane, and then looked back at plant cells only to find they had an extra rigid layer, the cell wall. Together, Schleiden and Schwann hit the nail on the head and hypothesized that all living things are composed of cells. But there was a missing piece to their puzzle. Where do new cells actually come from? Schleiden and Schwann kind of had no idea. Enter Rudolf Virchow in 1855. He delivered the final, perfected shape to the cell theory with this beautifully dramatic Latin phrase, omnis cellula e cellula, which means all cells arise from pre-existing cells. When old cells divide, new ones are born. This simple yet incredibly profound realization completed the cell theory as we know it today. Which brings us to Section 3, the universal cell components. We've talked about the theory, so let's talk about the hardware. If cells are the universal unit of life, there must be some universal truths, right? Well, let's uncover the components that apply across the board, from the tiniest bacteria to the trillions of cells sitting inside you right now. No matter how simple or complex, every single cell on Earth shares these three fundamental components. First up, the cell membrane. This isn't just a basic wall. It's a selectively permeable security gate. It decides exactly what gets in and what gets kicked out. Next, the cytoplasm. This is the semi-fluid arena filling the cell's volume. It's literally the chemical soup where all the metabolic reactions of life take place. Turn this off for even a second, and it's game over. Finally, ribosomes. These are the protein factories, and because they don't have a membrane, they're found absolutely everywhere, in both primitive and advanced cells alike. All right, Section 4, plant versus animal cells. Now that we know what all cells share, let's zoom in on the complex cells, the eukaryotes. Specifically, let's pit the plant cell against the animal cell to see what makes them so different. I love to use a brilliant little mnemonic trick here to remember these differences, the 4 C's. First, the cell wall, present in plants for rigid support but totally absent in animals. Second, the chloroplast. Plants have them to make food from sunlight, animals definitely don't. Third, the central vacuole. Now, in plant cells, this water balloon gets so massive it takes up like 90% of the space and literally kicks the nucleus over to the corner. In animal cells, vacuoles are tiny or just non-existent. And the fourth C? This centrosome. This is a special structure used for cell division that is present only in animal cells, not in plants. So just remember the 4 C's. Let's jump to section 5, prokaryote versus eukaryote. Let's step back for a second and look at the deepest duality in all of biology, the divide between the primitive and the complex, the prokaryote and the eukaryote. The names themselves actually give away the secret. Pro means primitive and eu means true. Karyon means nucleus, so a prokaryote has a primitive, naked nucleus with no protective membrane and absolutely zero membrane-bound organelles inside. It's kind of like a minimalist open-concept apartment. A eukaryote, on the other hand, has a true membrane-bound nucleus and specialized membrane-bound rooms, like the Golgi and mitochondria. Even their universal ribosomes differ in density. Prokaryotes have the lighter 70S ribosomes, while eukaryotes rock the much denser 80S versions. And this brilliantly illustrates a question of scale. When we say prokaryotes are primitive and small, just how small are we actually talking? Well, a typical bacterial cell is a mere 3 to 5 micrometers long. To put that into perspective, eukaryotic cells are typically 10-20 times larger than that. And if you want to talk about the largest single isolated cell in the entire world, that title goes to the massive ostrich egg. No way, right? But it's absolutely true. But listen, being tiny has a massive, frankly terrifying advantage: speed. E. coli bacteria have so little internal baggage that they undergo rapid, explosive, exponential cloning every 20 minutes. 1 becomes 2, 2 becomes 4, and within a single day you have billions. Contrast that with a complex eukaryote, like a human cheek cell dividing inside your mouth right now. Because it has to carefully organize and divide its massive nucleus and all those complex organelles, it has a super sluggish division cycle, taking a full 24 hours. The bacteria basically trade complexity for sheer, overwhelming speed. Section 6. Inside the bacterial cell. For our final chapter today, we are going to shrink ourselves all the way down and journey into the anatomy of a single, highly efficient bacterial cell to see what makes these prokaryotes tick. Now, most bacteria have a rigid, cement-like cell wall to keep them from popping, but there is a fascinating exception: mycoplasma. It's the smallest cell on Earth, measuring just 3 micrometers. And get this: it has absolutely no cell wall. Because it lacks that rigid outer armor, it can constantly change its shape. Biologists lovingly refer to it as the Joker of the plant kingdom, a shape-shifting prankster that totally defies the standard rules of bacterial structure. Deeper inside the bacteria, past the main tangled web of DNA, we find something truly incredible: plasmids. These are tiny, extra rings of DNA. They replicate entirely on their own and serve as brilliant survival tools. Plasmids carry bonus genes, most notably granting the bacteria resistance against our antibiotics. Today, modern biotechnology essentially uses them as gene taxis. Scientists can literally sneak a piece of human DNA, like the gene for insulin, into a plasmid and smuggle it right past the bacteria's defenses. The bacteria reads the plasmid, thinks it's its own, and just starts manufacturing human insulin for us. It's a biological hack of the highest order. And finally, you might be wondering, how does a bacteria breathe and function without the fancy mitochondria that we have? Well, the answer is the mesosome. The cell membrane simply folds inward upon itself, creating tubes and curves that drastically increase the surface area. And this folded membrane is the ultimate multitasker. It handles forming the cell wall, dividing and distributing DNA to clone itself, secreting materials, and essentially doing all the heavy lifting of respiration and energy production. It just goes to show, you really don't need a complex organelle if you know how to brilliantly fold your own outer boundary. We've seen today that from the simplest bacteria to the most complex human, the cell is the absolute required ticket to the living world. But this leaves us with a fascinating, kind of unsettling paradox. Viruses do not have a cellular organization. They don't have cytoplasm, they don't have a membrane, they are literally just genetic material wrapped in a tiny protein coat. By our strict biological definition, they are kicked out of the living club. They are not technically alive. So I'm going to leave you with this final provocative thought. If viruses aren't alive, how do these microscopic zombies hijack our cellular machinery and conquer the living world so effectively? That is a mystery for another day. Thank you so much for joining me on The Explainer. Keep questioning, keep learning, and I'll catch you on the next deep dive.